Hello, my college biologists. This is our first video of quarter 4. We are now viewing a transcription. So let us begin. Transcription starts outside of the cell when signals are received by receptor proteins. And these receptor proteins will isomerize, causing chain reactions in many other proteins. And that is what's being illustrated here in this sort of like domino effect. This protein is changed into this protein, which then changes 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 these proteins, which then, activate transcription factors. And so we finally got into the nucleus. We've got some proteins that can deal with DNA. So what happens with those transcription factors? Well, oh man, we should zoom in, shouldn't we? We should zoom in here. That's too much. All right. Here, at the beginning of a gene, here's the start of the gene, but around, I guess it's not exactly the beginning of the gene, it's sort of like a couple bases ahead of the beginning of the gene. But anyway, at the beginning of the gene, there is what is called the ta-ta box. And the wonderful transcription factors that we have just activated will be attracted to the ta-ta box. And transcription factors will continue to be attracted to the ta-ta box until they are able to attract the star of the show, RNA polymerase. Now that RNA polymerase gets himself set up at the start of the gene, but wait, he's just stationary. He can't do anything without the help of ATP. Oh, what did I just do? I'm supposed to zoom in and out on ATP. So with the help of ATP, polymerase is sent zooming down the gene. That is happening. Now, while it's zooming down the gene, it is momentarily disrupting the hydrogen bonds of the DNA. It is exposing the bonds of the DNA. So the two strands are separated from each other. They were forming hydrogen bonds to keep themselves together, but as the polymerase moves down, they get separated. And while they are separated, free nucleotides in the nucleus are attracted to the exposed bases of DNA. Well, how does that work? Nucleotides are made of a nitrogenous base, a sugar, and a phosphate. And these are, there's a thing that purines and pyrimidines do, and since nitrogenous bases are made of purines and pyrimidines, and they are found in nucleotides, the nucleotides can do this, and that is, when a purine will bond with a pyrimidine, it will bring the nucleotides together. So, how does that work? Let's get more details. Well, in those nucleotides, there are different types of purines and pyrimidines. The pyrimidines consist of uracil, thymine, and cytosine, and the purines consist of adenine and guanine. So how do they interact with each other? Well, adenine will bind with thymine in DNA, and guanine will bind with cytosine in both DNA and RNA, and in RNA alone, adenine will bind with uracil. Now this is kind of a little bit predictable, because if you look at uracil shape O, N, and the O and the N here are making the bond. If you go back to thymine, O, N, and the O and the N here are making the bond, it's basically the same shape. Thymine's got this extra little methyl group, the CH3 going, but other than that, it's pretty much the same as uracil. And so they both can form hydrogen bonds with adenine. So where does that get us? Well, as this occurs, I guess I'll go back to this one. As this occurs, the polymerase itself will form covalent bonds between the nitrogenous bases right here, forming covalent bonds between the nitrogenous bases. They're coming in to match up with the DNA on their own, but then the polymerase binds them together to create the mRNA polymer. And this will continue as the polymerase goes down the gene. You can see here the blue is the mRNA polymer that's sticking out. When it gets to the end of the gene, the polymerase will detach and release the mRNA, and you have reached the end of transcription. So let's review. Transcription is the process of creating RNA and occurs in the nucleus. First, receptor proteins detect external signals, starting a chain reaction of many proteins, leading to the activation of transcription factors in the nucleus. Then, transcription factors are attracted to the ta-ta box at the start of the gene. RNA polymerase is attracted to those transcription factors, and several ATP donate phosphates to the RNA polymerase, triggering its movement down the gene. As RNA polymerase moves, it momentarily unwinds the DNA double helix by disrupting hydrogen bonds between the nitrogenous bases. The nitrogenous bases of free nucleotides are attracted to the nitrogenous bases of the newly exposed nucleotides of the DNA. This works because some purines form hydrogen bonds with some pyrimidines. Adenine and guanine are purines, while cytosine, uracil, and thymine are pyrimidines. In DNA, adenine will form hydrogen bonds with thymine, and in RNA, adenine will form hydrogen bonds with uracil, and in both DNA and RNA, guanine will form hydrogen bonds with cytosine. As RNA polymerase travels down the gene, it forms covalent bonds between the phosphate of one free nucleotide and the sugar of another to create the mRNA, which is known as messenger RNA, because it sends the message out of the nucleus. The DNA recoils as RNA polymerase travels, and when it reaches the end of the gene, the mRNA is released, and the RNA polymerase detaches from the DNA. Thank you for being patient with me. I hope this helps. Struggle well.